Now, before we start to understand peptides and how they work and how they're formed, it's really important for us to understand how two specific functional groups can bond together. So peptides are developed from lots and lots of amino acids which join together. Now we know that amino acids all have a carboxyl functional group and an amino functional group. And so if we want amino acids to bond together, then the best way for this to happen is for the amino functional group of one amino acid to bond with the carboxyl functional group of another amino acid. And so this can happen quite easily via a condensation reaction. So if we have our amino group and our carboxyl group, then what happens when two amino acids bond together is that a condensation reaction occurs. So we lose the OH group, the oxygen and the hydrogen atoms from our carboxyl functional group, and we lose one of the hydrogen atoms from our amino group, and they split off to form a water molecule. So what we have left is we get a bond between the remaining carbon atom and the nitrogen atom. And so usually the way that we represent this is like this. So that is what we are left with. We're left with this, the carbon double bonded to the oxygen still, and the nitrogen still bonded to one hydrogen, but now the carbon and the nitrogen have formed a covalent bond together. And so here we've got the rest of our first amino acid, and here we've got the rest of our second amino acid. And so what we call this linkage here, this linkage that can form between two amino acid molecules, is we call it an amide linkage. And so amide linkages are very, very important in the formation of peptides. They are produced through a condensation reaction between a carboxyl functional group and an amino functional group. Now, when we're bonding amino acids together, we have specific terms that we can call the resultant molecules. So here we've got two amino acids bonding together via one amide linkage. And we can call this, when we have two amino acids bonded together, we call that a dipeptide. If we were to have three amino acids all bonded together, if we were to have this arrangement, so two peptide linkages bonding three amino acids together, then we get a structure that looks like that. And simply we call that a tripeptide, meaning three amino acid molecules bonded together into one chain. And so as we get more and more amino acids bonded together via amide linkages in a long chain, we call these, the resultant molecules, we call these polypeptides. And so once we get very long polypeptides containing, roughly 50 amino acids bonded together or more, then we can call these proteins. So proteins are simply just long strings or long chains of lots and lots of amino acids bonded together. And so that is what our peptides are. We're simply dealing with these amide functional groups joining multiple amino acid molecules together. Now, obviously we call this setup, this kind of linkage or this functional group here, an amide linkage. Now, however, if this linkage is occurring in a protein, then we have a more specific name for it. Obviously, an amide linkage can occur in lots of other substances that aren't organic substances in the body or that relate to proteins. We can form amide functional groups in lots of other more inorganic or sort of more industrial processes. However, when we form an amide linkage in a protein, we can then refer to it as, rather than an amide linkage or an amide functional group, we can refer to it as a peptide functional group or a peptide linkage. And now that we can only use the word peptide when our amide functional group is in a protein molecule. So we're going to have a brief look at a bit of an example now. We've got two amino acids down here. We've got glycine and we've got threonine. What we're going to do is we're going to write a chemical equation for the condensation reaction between glycine and threonine. And we're going to draw the resultant dipeptide that is produced. So if we go up here, we will figure out the molecular formula of each of glycine and threonine, and we'll be able to quite simply and quite straightforwardly write a chemical equation. So if we have our glycine here, then we can see we've got 2 carbons in our glycine molecule. We've got 5 hydrogens, we've got 2 oxygens, and 1 nitrogen. And so the molecular formula of threonine, we have 4 carbons. We have 1, 2, 3, 4, 5, 6, 7, 8 hydrogens. We have 3 oxygens, and again 1 nitrogen. Now we know that in this reaction it's a condensation reaction, as we're forming an amide linkage between our 2 amino acid molecules. So we know that through this condensation reaction we have 1 water molecule being split off. So we can write that in here. And now obviously the resultant dipeptide that is produced by the bonding or the condensation between these two molecules is going to contain all of the atoms in these 2 amino acids, with 2 hydrogens less and 1 oxygen less, as those 2 hydrogens and that oxygen have disappeared and gone into a water molecule. So that means that we can simply subtract these from the net number of atoms over here. So we've got 6 carbons. On the reactant side of our equation, we've got 13 hydrogens less 2 gives us 11 hydrogens. We've got 5 oxygens less 1 gives us 4 oxygens. And finally 2 nitrogens. So that is the molecular formula of our resultant dipeptide. Now, if we want to draw the full chemical structure of this dipeptide, it's simply a matter of converting the carboxyl functional group of one of the amino acids and the amino functional group of the other amino acid and joining them together via an amide linkage. So here we have our glycine. So everything else in the molecule is remaining the same. We're simply changing the 1 carboxyl functional group in this case. There are in fact 2 different ways these could bond together. What we're about to go through is the bonding between the carboxyl functional group of glycine with the amino functional group of threonine. But in fact, we could have the carboxyl functional group of threonine bonding with the amino functional group of glycine. But in this case, we're going to deal with these 2 here. So this is the stem, and we know that we're losing this OH. We're losing that's being split off. So we're left with the C double bond O. We know that is going to bond straight to the nitrogen, like this. And this nitrogen is now only bonded to 1 hydrogen because one of the hydrogens has been split off. And again, the remainder of the dipeptide molecule remains the same as the amino acids that we used to produce it. And so this is what our resultant dipeptide molecule looks like. And that is the dipeptide produced by the bonding of glycine and threonine, complete with our amide linkage in the middle here, our amide functional group. And so that is how amide functional groups are very important in the formation of peptides and eventually proteins. And so we'll go on to look at a little bit more about the properties of our larger protein molecules in another video.